Hello students, today I am going to start a new chapter which is genetics. In the previous class chapters we have studied about the reproduction in organism, then in plants and all. In genetics we'll study about the new terms like pleiotropy, polygenic inheritance, complementary genes, and a brief description about Mendel's experiment and the various terms used by Mendel. Brief description because I have already explained this chapter for class 10th in my previous video for class 10th. So you can refer to the video for the detailed description about all the factors. Today just a brief account for all these terms and the new terms in this chapter. This we'll study in detail. So let's start with the term Mendel's experiment. Mendel is the father of genetics because he was the one who started that the term genes. Genes was not coined by Mendel. He has used the term factor. That some factors are present in the offsprings which are inherited from the parents, from the grandparents, or that is the factors are inherited from generation to generation. But nowadays it was named genes. Genes are given to this, and according to Mendel, these genes are present on the chromosomes. From the chromosomes they are inherited to the offspring and to the further generation. But there is a sequence for inheritance, and according to his studies he has according to his studies he has given up rule for this, or the laws for this experimental study, or this because pea plant, he has taken pea plant as he has selected pea plant because for the reason that it is having contrasting characters like the tall and short plants, then flower color. I'm just giving a brief account because I have already explained all these things. So the contrasting characters, the lifespan of the pea plant to get the next generation is very short, and it is easy to handle. That's why Mendel selected pea plant as his as for case study. And according to him, the characters are transmitted from one generation to another, and some of the characters which may be considered as dominant characters are expressed in all the offsprings of F1 generation. The first generation observed was given the name first filial generation. In the first filial generation, the characters are from the parental character, one of the parental character, and whichever is the dominant one. After the self-cross of the first filial generation, the second filial generation gives a ratio of 1:2:1 as a genetic composition genotype, while the phenotypic composition is 3:1, where 3 is for the dominant one and 1 recessive. It means recessive character expresses itself in the second filial generation. Recessive character is the one which remains hidden in one generation and expresses itself in the second filial generation. According to these experiment, he has given 3 laws: law of dominance, law of segregation, and law of independent assortment. We'll study all these terms with an example. Next term is Mendel's inference. Mendel's inference, I have already explained that there are factors present on the gene, present on the chromosome. These factors are inherited from parents to offspring. These factors are responsible for the physical expression or the phenotypic expression of an organism, of a plant, or of a part of a plant or an organism. So that phenotypic expression or the physical expression is because of the factor which is the gene present on the homologous chromosomes, or the gene which is responsible for the contrasting characters. Contrasting characters are termed as alleles or allelomorphs. So allelomorphs, contrasting character, one may be the dominant one and the second one is the recessive. So let's start with the Punnett square. We have taken an example of tall plant and dwarf plant. Tall and dwarf. And this tall is represented by capital T, both capital T, and dwarf with small t, both small one. Then the capital T and small t is the gametes. Gametes for first generation. Here, by the fertilization, the fusion between two results in the first generation, which is the F1 generation. F1 generation is having capital T and small t and all tall plants. All tall plants. It means tallness is the character which is dominant for this plant. So dominant character is expressed in the first generation. After the self-cross between two tall plants, the gametes for the F2 generation are written in a square pattern. Capital T, small t, capital T, small t. These are the gametes for F2 generation. And when we are making a cross between this, we are getting one capital T, two capital T, small t, and one small t. It means one tall, two tall, one dwarf. But the first one is homozygous tall, while the second one is heterozygous tall. It means heterozygous condition, the alleles are from both the parents, and the third one is homozygous dwarf. Now always it is asked what is the genotypic expression or phenotypic expression. Genotype is this T, capital T, small t represents the genotype. So genotype is 1:2:1. Genotype 1:2:1. Capital T, 1 capital T, both capital T, 2 capital T, small t, and 1 both small t. So this one, and the phenotype is 3:1, 3 tall and 1 dwarf. So this is the phenotypic expression. Next, Mendel's law, according to these cross, according to Mendel's first law, which is the law of dominance, the character which expresses itself in the first filial generation is the dominant character, while the one which remains hidden is the recessive character. Based on that, in this case, the character which is expressed in the first filial generation is the tall character for tallness, and that tall character is the dominant one. So while the dwarf characters remain hidden, the alleles for dwarfness remain hidden. So this one, the tallness is the law of dominance. At first generation, the dominant character is shown in the generation, while the other character, the recessive character, remains hidden. Again, the law of segregation. According to the law of segregation, that the genes are not blended or mixed with each other, they remain segregated or separated, and in the further generations, in a ratio 1 or 1:2:1 ratio, they are expressing their characters in this ratio. They are not blended. So blending of characters, segregation can be proved by this one. Like we can see capital T represents the dominant character, small t for dwarf. Here Both capital T gives the expression for tall, capital and the T in the heterozygous condition, which is capital T and small t, gives tallness, but in a heterozygous condition. Here we can say that they may be blended because the genes mix to give tallness, but one recessive presence of one recessive character shows or proves that the characters or the genes are not blended. The genes are not blended, they remain segregate. They can they can express themselves in any of the generation, as they are expressing even in the ratio 1 ratio out of the 4 progenies getting we are getting in this square. Out of 4, they are expressing their character in ratio 1, which is that they may express. So when the condition is they may express their characters, it is the law of segregation. Then independent assortment. Independent assortment is they are segregated, they are separated, they show their characters independent of each other, like capital T for dominance, small t for recessive in the second generation. Then the mixed or the heterozygous condition again gives the dominant, but they remain separate, and each character is able to express its original character, which is the tallness or dwarfness in the homozygous condition. So that is the expression given by its the expression, the original expressions by the genes is the law of independent assortment. Next one, but there are some exceptions for this, like incomplete dominance or codominance. And this is an example of incomplete dominance in case of Mirabilis jalapa or 4 o'clock plant. In this, the flower, we have here there is an example for red colored flower crossed with white colored flower, where red is the dominant character while white is the recessive one. When these two are crossed, the gametes are capital R and small r, and in the first, these are the gametes, and in the first generation, this should be a red colored flower according to the Mendel's law, according to the Mendel's expression or the experiments. But in this case, the first parallel generation, the F1 generation, is a pink colored flower, which is an exception for Mendel's character. Pink colored flower, after the self cross, the characters expressed are for the R, capital R, small r, taken in a Punnett square. This capital R, when it is present in dominant in the homozygous condition, means 2 capital R homozygous condition, then the flower color is red. If the R is in small letters or it means homozygous condition, the flower is white color. But when it is present in heterozygous condition, the color is not of the dominant color, it is a blended expression of red and white, which is pink color. So this is an example of incomplete dominance, where the dominant character is not completely dominant over the other or the recessive character. So the expression is a mixed expression. Next is codominance, which is an exception for the Mendel's experiment. Codominance, as in case of blood group, which is O, A, B, AB. Here it is the genes are represented by I, and the characters like I, O is for homozygous O blood group. But if the character is in this way, like heterozygous as well as homozygous condition, so then it is for blood group A, various genes or contrasting characters are present and they express themselves in the presence of its complementary or the other partner. So this is an example of codominance. Next is next let's come to the today's topic, or all the topics we have, previous topics we have studied. One more thing, the test cross. Test cross is a cross to detect that the characters are parental characters dominant from the dominant parental characters or the recessive. In this case, the plant or the genotype is crossed with a recessive gene, homozygous recessive gene, to detect the presence of parental character, or the homozygous dominant or the homozygous recessive ones. Let's start with the today's topic, pleiotropy. Pleiotropy is a condition in which a single gene is responsible for various characters, like in case of plant, the starch synthesis or the starch in the grains or the size of the seed. All these characters are given or expressed by a single gene. So in this way, if a single gene is responsible for various phenotypic expression, then the phenomena is termed as pleiotropy. For example, here color of the flower or the seed coat, different phenotypes, but the expressions are given by a single gene. This is a case of pleiotropy. There is a PKU, which is a defect, which is this defect is termed as phenylketonuria. Ketonuria. This is a defect or the phenotypic defect. This defect is due to an enzyme, lack of an enzyme, which the enzyme which is responsible for the conversion of phenylalanine into tyrosine inside liver. In liver, the absence of this enzyme results on lack in conversion of this phenylalanine to tyrosine, results in various phenotypic expressions like mental retardation, eczema, or depigmentation or pigment loss in the skin. So all these factors are the effect of mental retardation, eczema, and pigmentation loss is due to the lack of the enzyme responsible for this conversion. And then one more example is for the Drosophila. Drosophila, in case of Drosophila, white-eyed color, white eye, the gene responsible for white eye, gives various depigmentation in the body parts. Depigmentation in the body parts are controlled by the gene responsible for the white eye color. Next, in case of transgenes where the genetic composition is changed, introduction of gene results in various other phenotypic changes in the organism. So that is also a pleiotropic factor. There is polygenic inheritance. Polygenic inheritance is that if a dominant gene is totally or partially able to express itself, then it is a polygenic inheritance. This is given by Galton. In this, that suppose for skin color, which melanin is responsible for the skin color in human. If there are three factors responsible for this pigmentation, if all the three factors are in the are present in a homozygous condition, homozygous condition which is all dominant, then the color is very dark for the person. If the alleles are in homozygous recessive condition, then the skin color is very light. If they are present in a heterozygous condition, the expression is an intermediate expression. Intermediate expression which is termed as mulatto. Next is the complementary gene. Complementary gene is the presence of two non-allelic genes which are dominant, but without the presence of other, they are not able to express themselves. Non-allelic dominant gene giving up dominant phenotypic expression, but absence of the other or the complementary results in another phenotypic expression. For example, if the C and P are the genes or the alleles responsible for the color of the flower, if the C is in homozygous dominant condition and P in the homozygous recessive condition, in this C in the recessive and P in the tall, both capital means dominant, then if one of the gene is dominantly expressing and other is recessive, then only the white color is expressed. If C is both capital and P is both small, then only white color is expressed. If both P is capital and C is either capital or small, means out of C and P, one must be in both capital, then only the expression for white colored is shown. In this, as you can see, in the first filial generation, the genotype is capital C, small c, capital P, small p, which gives a colored flower. This is in case of Lathyrus odoratus, which is a pea species of pea. So pea plant Lathyrus odoratus, the color, if the genes are present in a heterozygous condition, the expression is colored flower. There is a self-cross between the colored flowers, and the alleles or the gametes are capital C, capital P, capital C, small p, then the other one with small c, capital P, small c, small p. Again here also the same gametes. When we make a cross between this, if the C or the P are present in both dominant one, both capital C or both capital P, other maybe a capital or small, then only the white colored flower is shown. Like here, capital C, capital P, the flower color is white. Here capital C, capital P, small p. The other is a recessive one. Thus other must these two are present in a capital form or the dominant form, then the white colored flower. So in this way we can make this Punnett square. If the composition is like this anywhere, then the flower color would be a colored flower, and the ratio what we get is 9:3:3:1 based on the colored and white flower. So this is all about today's topic. In the next topic we'll study about the defects and other factors or other terms, new terms for this chapter. Thank you for today's session.